So the next topic we're going to move into for a while here is genetics. And in lab, the last couple weeks, you've been doing some stuff with fly genetics. So you are familiar with some of the basics and some of the terminology. So I encourage you to not forget about that as we're talking about this in this part of the class as well. So first, again, the father of genetics, if you want to call him that, is Gregor Mendel, who was a monk in Austria, and his organism of choice were the garden peas, or Pisum sativum, that's shown in this slide. And I don't want to spend a lot of time going through the basic stuff, but I think it's important to start this from the point of experimentation. And so Gregor Mendel, back in 1865, he was working with these peas, and he had the ability to breed them for lots of different traits. And he was particularly looking at 7 different traits in these peas. And he was able, through breeding in his gardens, to generate what we call true breeding strains. These are true breeding or pure breeding, it's the same word. These tend to be homozygous, and I know you're familiar with that word from what you've been doing in lab. And he was also able to generate hybrids of these plants, which would be the heterozygous versions. So this slide just shows you the 7 different traits that he was investigating. And what you notice is that they're discrete traits. In other words, flowers were either purple or white. There wasn't anything in between. And at the bottom, some of the plants he was looking at were either tall or dwarf, round or wrinkled peas. So all of those, and that was important because he set this up to really have some nice traits that he could really easily assess and analyze using his simple organism, these peas. So let's look at some of the experiments So this slide, I'm just showing you a summary of the data that Mendel actually collected. And what you notice here, the previous slide we were looking at tall and dwarf, and so that's at the bottom of this table. But these are all the different traits that he was looking at. And what you notice in that far column that says ratio is that in nearly every case, well, in all the cases shown here, the ratio was roughly a 3-to-1 ratio of the dominant trait, which is the one that he saw in the F1. That's how we define what the dominant trait was. It's whatever trait you see in the F1s. And that the dominant trait was present in a ratio of 3-to-1 in the F2. And so these were the data that Mendel gathered by doing a number of these crosses. And just to quickly remind you, and you've done this, I know, in lab too, and many of you have done this before. So this is a Punnett square, and this is just the way that we now, as we think about what's going on with some of Mendel's experiments, this is how we keep track of that random fertilization. And so across the top, this is showing male and female, but of course, we're doing this in plants, but there are male and female gametes. So across the top, if we're doing an F1 into the F2, you write the possible gametes from the male, across the top, the possible gametes of the female along the side, and then all the squares inside are all the possible combinations that you can get. So a Punnett square is really a mechanism or a tool that we use to account for all the possible random fertilization events. And hopefully you're familiar with this tool as we move forward. But what I want to emphasize here is the results that Mendel acquired in these experiments were what really illustrated what we now know as Mendel's law of segregation. And that law of segregation is basically described here, but it's that 2 copies of a gene or 2 different alleles, right? We've defined those, and there's also a lot of genetic terminology that's defined nicely in your textbook. So I would refer you to some of the textbook pages if you're still unsure about some of these terms, because we're going to be using them a lot, and so make sure you understand what they mean. So back up. Mendel's law of segregation means that the 2 different alleles of a gene, they segregate from each other at every generation. And so you see that here in this diagram. So we have the heterozygous F1 generation, And during meiosis, right, the gametes form, and then in that Punnett square we saw in the previous slide, we put all the possible combinations together and we come up with all of those possible genotypes that are shown there in the F2 generation. And again, a Punnett square is really about organizing all the possible random fertilization events. So where do we know and understand now that Mendel's law of segregation comes from? At the time, remember, he did not know what meiosis was, but we now know and understand where that comes from. And before we look at another figure about meiosis involving that, just want to remind you what we're looking at here are two homologous chromosomes, one in blue, one in red, and we use the term locus to talk about a specific location of a gene on a chromosome. And remember, homologs, one of these came from one parent, the other one came from the other in a diploid. And so a locus is a specific site where a gene is located, and homologous chromosomes are going to have the same genes at the same locus or the same location. But remember, they don't have they can be different versions of the gene. And in this case, we're talking about on one of the homologs in a heterozygote. On one of the homologs, there'd be an allele that codes for tall, and then on another homolog there would be the other allele that codes for dwarf. So clearly this is a heterozygote. Certainly if it was a homozygote, we would maybe have, the big T or the tall allele on both homologs. Or if it was homozygous recessive, we'd have the two little t's or the dwarf alleles on both homologs. And so this is where this comes from. So now let's take this and think about what this means and what Mendel's law of segregation means as we think about it in the context of meiosis. So again, if we remember how meiosis works, this is why it's really important to make sure we understand that. It really helps us follow inheritance and genetics. So when we look at two homologs shown in the top here, in this case we're looking at a heterozygote, remember, during meiosis 1 is when those homologs separate from each other. And if you have the recessive allele on one chromosome indicated in red here and the dominant allele on the other homolog indicated in blue here, during meiosis 1, those two separate from each other. So Mendel's law of segregation, that two copies of a gene separate from each other at every generation, we know that that's because homologous chromosomes separate from each other during anaphase 1 in meiosis, and that's really nicely illustrated in this slide. So before we leave the topic of monohybrid crosses or inheritance of a single gene, there's one other important type of cross that I wanted to bring up because this comes up a lot, and if I use the term test cross ever in a quiz or on an exam or when we're talking, I just want to make sure you know what it means. And so when we have, in this case as shown in this slide, when we have a tall plant, that tall plant could be homozygous for the dominant allele or it could be heterozygous. And there's no way in this particular situation for us to determine what the genotype is when the plant has the dominant phenotype. So a test cross is a way that you can determine the genotype in this case. So a test cross always involves the organism that you have in question or the one you're studying, and it crossed by a recessive organism. Because we know the genotype of a plant with a recessive phenotype, right? I mean, we know that must be homozygous recessive. So if we cross a plant that shows the dominant phenotype to a dwarf plant in this case, we know that dwarf plant must be homozygous recessive. If the plant in question, the one showing the dominant phenotype, is homozygous for the dominant allele, we would get the results shown on the bottom left. And we'd get this ratio, or we'd get all of the plants, I should say, heterozygous and all showing the dominant phenotype. Alternatively, if the dominant phenotype plant at the top is the heterozygote, and we did a test cross with that, in other words, crossed it with the homozygous recessive, we would get a different ratio. We would get a 1-to-1 ratio of tall plants to short or dwarf plants. And so a test cross is a way for us to use to determine what an unknown genotype might be. So in a test cross, one of the parents or one of the organisms involved is always the one that has the recessive phenotype, because we know the genotype of that particular organism. Okay, so now let's start talking about crosses that involve not just a single gene, but crosses that involve two genes, or in other words, what's called a dihybrid cross. And you see similar diagrams to what we'd been looking at before, where we have a P generation. In this case, again, P generation is pure breeding or true breeding parents. And we've introduced here not just, what we've introduced here now is the seed color and shape. And so the dominant trait for seed color is yellow, and the dominant trait for seed shape is round. And so then the recessive is green and wrinkled. And so we're looking at the inheritance of both of those traits in one particular cross. And if you think about this, there are a couple of different hypotheses that could be tested in this particular case. So if we have two pure breeding plants at the top in the P generation, and their genotypes are shown there, during meiosis in the formation of the gametes, we have the two dominant alleles, because that's all there is in one of the parents, and the two recessive alleles, because that's all there is in the other parent, coming together at fertilization to form the doubly heterozygous organism in the F1. And again, remember, this is how we know that yellow is dominant, because that's what we see in the F1, and this is how we know that round is dominant, because that's what we see in the F1. But when we think about what happens in the F2, there are two different hypotheses that we could actually test. And one of them is shown here on the left. That's the hypothesis of linked assortment. And that would be where the alleles in the parent always stay together. In other words, in this case, the two dominant alleles, the big Y and the big R, always segregate together through the F1 and into the F2. And then the two recessive alleles always segregate together. And you see what that might look like in that Punnett square there. So the two dominant alleles always go together in the one parent, and the two recessive alleles in the other. And in that case, then, you would expect to see this, again, a 3-to-1 ratio of yellow and round, 3/4 of those, and green and wrinkled. And so that's what we would expect to see if the hypothesis of linked assortment was supported. But in the other panel, we see another hypothesis that we could test, and that is the hypothesis of independent or unlinked assortment. And so if we follow that from F1 down to F2, instead of the parental alleles staying together, this hypothesis suggests that the parental alleles are randomly distributed. In other words, there's nothing that holds the dominant allele Y and the dominant allele R from segregating together all the time. So you see at the top of the Punnett square there at the bottom, you see all possible combinations. We could get both dominant alleles together in a gamete. We could get the dominant for color and the recessive for shape. We could get the recessive for color and the dominant for shape, or both recessives. And so you see all possible combinations that would indicate that they would randomly assort. And so Gregor Mendel did some experiments to actually test this hypothesis, and so we'll look at the results that he actually got in the next slide. So here are the results that Mendel actually got in his experiments, and what we see is not that 3-to-1 ratio that supported the first hypothesis of linked assortment, but instead we see ratios and numbers that support the second hypothesis, that second panel on the previous slide. And that is a 9-to-3 to 3-to-1 ratio with all the possible combinations, right? You can have yellow and round or yellow and wrinkled, etc. But again, you can see in the F1 both dominant traits showing up there. And so after he carried out these experiments, this led to the his putting together what we call Mendel's law of independent assortment. And that is that alleles of different genes assort independently. They don't assort in a linked fashion, they assort in an unlinked fashion. And so where does this come from as we now understand more about meiosis? We'll talk about that in the next slide. So if we dig in and look at meiosis, again, carefully, what we remember, we've got two different options shown here in the two different panels of meiosis. So once again, you have now not just one gene locus shown here as was in the previous meiosis diagram that we looked at a little bit ago, but now we're looking at two genes, right? We're looking at the gene at the Y locus that determines seed color. And we're also looking at a gene on the R locus that determines seed shape. And so you see, this is again, these are both heterozygous F1s, and you see that at the top. And we remember at metaphase 1, the homologous chromosomes actually line up opposite one another, and this whole process is random. So in the middle panel here, we see metaphase 1 can occur in different ways. We could have the red chromosomes all on one side, you could have, and then the blue would be on the other, or you could have any mixture of red and blue on either side. But again, we know at meiosis 1, those homologs separate from each other, but how they line up determines which alleles are going to go together in the end meiotic product. And so we know Mendel's law of independent assortment is due to the random assortment of homologous chromosomes at metaphase 1. So in any meiosis, homologous chromosomes can come together in metaphase 1 in various combinations, which is what we really refer to as independent assortment. So now I want to talk a little bit about a probability rule that we often use to make calculating some of these probabilities a little bit easier. So the product rule specifically states that the probability of two independent events occurring at the same time is the product, or you would multiply them. It's the product of the individual probabilities. So to think about this, let's look at this example. So if we have a cross indicated there at the top, so we have a double heterozygote crossed with an organism that's heterozygous at T, but homozygous recessive at Y. So because of independent assortment and these two things occurring independently, we can just consider T by itself first. Let's ignore Y. And we can consider T. And if we just consider T, we would get the Punnett square shown there on the left. And we can do the same thing then for Y. We can ignore T because Y is independent of T. And if we crossed an organism with the Y genotype indicated, we would get the Punnett square that's shown there on the right. So now let's use the product rule to ask a specific question. So if we wanted to figure out what, in doing this cross that's indicated here, what's the probability of getting an organism that's dominant, homozygous dominant at T, but homozygous recessive at Y? Well, what is the probability of being homozygous dominant at T if we did just that T cross? Well, that would be 1/4. It's 1 out of the 4 squares. And then we can ignore T, and what's the probability of being homozygous recessive at Y? Well, from the Punnett square, we can say that's 1/2, or 2 out of those 4 squares. And so what the product rule allows us to do then is to multiply those two probabilities. So the probability of getting homozygous dominant at T, homozygous recessive at Y, is 1/4 times 1/2, or 1/8. So now let's use this to go back and look at the results that Mendel got that we looked at earlier. So here again you have that Punnett square, his results supporting that law of independent assortment. So what we've done here is the cross with heterozygous at Y, heterozygous at R, and it's a self-fertilization. So if we wanted to figure out, using the product rule, the same data that's shown in this Punnett square, one would ask first, what is the probability of having the dominant allele at both genes? So this could be homozygous dominant or heterozygous. And so if we think about, let's just look at the Y cross, again, ignore R, let's just look at Y. but what's the probability of being homozygous or having the dominant Y allele? It doesn't matter what the other one is. Well, that would be 3 out of the 4 squares, or 3/4. And the same thing for R. So what's the probability of being dominant at both? That's 3/4 times 3/4, or 9/16. Then you can work your way through all of these. Well, what's the probability of having the dominant allele at Y and then homozygous recessive at R? Well, that's 3/4 times 1/4, or 3/16. And then same 3/16 when you're homozygous recessive at Y and dominant at R, and then homozygous recessive at both would be 1/4 times 1/4. And so this ratio is our 9 to 3 to 3 to 1 ratio. So you can use the product rule to get the same data that you could get by drawing out this whole Punnett square, because if you count all of these up, you will see that there are 9 squares that are yellow and round. There are 3 squares that are yellow and wrinkled. There are 3 squares that are green and round, and then there's 1 square that's green and wrinkled. Okay, so now let's just take this one step farther. So let's say we have the cross that's indicated here now, where we're looking at 3 genes, A, B, and C, and we set up this cross. And we ask the question, in this particular case, we're asking the question, what's the probability of getting the dominant phenotype at 2 of these 3 genes and the recessive phenotype at the 3rd? So the first thing you have to think about is that there's more than one way to do that, right? You could have the dominant phenotype at A and B, but then recessive at C. You could have dominant at A and C, recessive at B, etc. So there's more than one way of fulfilling this question. And so take some time to figure out what is the probability of each of these by using the product rule. What is the probability of getting the dominant phenotype at A, the dominant phenotype of B, and the recessive phenotype at C? And use the product rule to come up with each of these probabilities. Then once we do that, we would actually, in this case, after we've calculated the individual probabilities of each of these possibilities, is then we would add those probabilities together, or in other words, apply the sum rule. So what the sum rule is about is that the probabilities of mutually exclusive events is the sum of the independent probabilities. So in other words, the first scenario there, where we have the dominant phenotype at A, the dominant phenotype at B, and the recessive phenotype at C, that can't happen at the same time that the second option happens, right? These are just 3 different ways that fulfill the question or answer this question about being dominant at 2 genes and recessive at the 3rd. But they're mutually exclusive. In other words, all 3 of these things can't happen at the same time. So then we would apply the sum rule, where we would add those probabilities together. So the last thing I want to talk about here is sex linkage. And to do that, I want to talk about some original experiments that really put this idea forward. And so if we go back and think about the 2 laws that Mendel put into place, the law of segregation and law of independent assortment, we can trace those now to chromosomal movements in meiosis. We know and understand where those come from. And so we know genes are on chromosomes, and all chromosomes, of course, include sex chromosomes. So sex linkage, or sex-linked traits, was first uncovered in fruit flies by a researcher called Thomas Hunt Morgan. And so here's sort of an overview of his experiment, and this was done in 1910. And there was a rare white-eyed male that they discovered. So fruit flies' normal eye color is red, and so having a white-eyed fly is sort of unusual. And Morgan's experiments, again, were the first to really demonstrate this correlation between a particular trait or characteristic and the inheritance of sex chromosomes. So we can see in this slide the experiments that Morgan carried out. So we have this rare white-eyed male, and he crossed it in a P generation with some red-eyed females, and got an F1 generation that were all red-eyed flies. And then he took some of the males from that F1 generation and crossed them with females from the same generation. And when he looked at the F2, he saw some very unusual results that are shown at the bottom of this slide, but let's look more closely at the data that he collected. So what was noticed is that there were no white-eyed females in the F2s when you started with this original strange white-eyed male. And so what could be the cause of this? And it's showing again this correlation between a particular trait and the sex of an organism. And there were no white-eyed females in the F2. So if we take this information and put it into our tool that we've been using to follow things, or a Punnett square, let's look at what that looks like. So the F1 male, let me first clarify some nomenclature here. So males are XY, females XX in the fruit flies, and the normal allele, the one that gives red eyes, is indicated with a W+. This is just how fruit fly geneticists do it. And so a normal red eye allele is W+, and that's the dominant allele. And then the white-eyed allele, the sort of odd, strange version of the gene, is indicated with just a W. So if we took an F1 male that was red-eyed, if you go back to the 2 slides ago, you can follow that this was the genotype of the F1 male. So then we write the gametes across the top of this Punnett square, because remember, X and Y act as homologs during meiosis. And so we'd have half of the gametes with the X chromosome carrying that normal W+ allele, and then half of the gametes carrying the Y chromosome. And then the female, in this case, the F1 female, was heterozygous. So she had the wild type W+ allele and the mutant W allele. So if we do that and then fill in the Punnett square, what you can see is that there's no way to get a white-eyed female out of this cross. And so we now know that the gene for white eyes is actually carried on the X chromosome in fruit flies. And so there are a number of genes on the X chromosome. So all of those traits that are associated with genes on the X chromosome are what we would call sex-linked traits. And this is what we would call sex linkage. And so there's a number of these in humans too. So let's just look at one example of that. So in this case, we look at this is hemophilia, which is a blood clotting disorder in humans, and the gene that's involved in that disorder is on the X chromosome. And so in this particular case, we're taking a male. So in this example, the capital letter H as the superscript is the normal version of this blood clotting gene. And the little lowercase h with the /a, that's the recessive or the mutant allele, the one that actually causes this abnormal blood clotting that you see in hemophiliacs. So we take a normal male with normal blood clotting, and he has children or mates with a female who's a carrier for hemophilia. And as we put the Punnett square together, these are all of the possibilities that could come out of that. And so the females, one would be normal, the other would be a carrier, so neither of them showing the hemophilia trait. And in the males, we'd have half of the males normal, but the other half of the male offspring would have the hemophilia trait. And so this is an example of sex linkage in human disease. There are a number of other examples besides hemophilia. Color blindness is another one that's commonly discussed in genetics, and that one is also associated with the X chromosome. So I want to now move on and talk about pedigrees and how they are used to help us determine the mode of inheritance of certain characteristics or certain disorders. And before we do that, this slide just shows you some of the basic symbolism and how we refer to pedigrees. And so a pedigree, of course, in humans, they're like family trees showing the different generations. And we can look at them and indicate whether something was inherited in an autosomal fashion, meaning not sex-linked, the gene is on one of the non-sex chromosomes, and whether that's dominant or recessive. Or we can also determine, often by looking at pedigrees, whether it has a sex-linked mode of inheritance. So just look at this slide and familiarize yourself with some of the typical symbolism and how we indicate males and females Females circles, males with squares, and then affected individuals are usually filled in. And then we have other symbolism that we use for heterozygotes or people who are carriers. So just for fun, I just want to show you this pedigree. I always like following the British royal family, so this one is kind of interesting. So this is a very large pedigree, dates back to, the 1700s. And this, you can see on the far left side, it really involves the current British royal family. But you can see a number of filled in squares on this pedigree. So hemophilia did segregate in descendants of our current British royal family. And I have a few people that you may be familiar with indicated there in red arrows. And so the current Queen of England is shown there, Elizabeth II, and her son, Prince Charles, is there. And then their two sons, William and Harry. And of course, we know that this pedigree isn't complete now because Prince William and Kate have now 3 children, and Prince Harry and Meghan had just had Archie. But what I want you to know a couple of things about this. First of all, the current British royal family has escaped any chance of hemophilia, because if you look back where Elizabeth II is indicated, her father, George VI, did not have hemophilia, nor did her mother isn't indicated on here, but because he did not have hemophilia, none of his descendants then would, of course, have it, because it is on the X chromosome. And a couple of other things I just think are fun from this slide. So there are a number of really great movies and other shows out. Some of you may be familiar with The Crown, which goes back and tells the history of the British royal family. And then currently, I think Sunday nights on CNN, there's also The Windsors that I've been watching. But there are some other really good movies that some of you might be interested in watching during this downtime. The Madness of King George is about George III, which is shown way up there at the top of this pedigree. And then The King's Speech, Colin Firth is the lead in that. And that is a story about actually the current Queen's father, who was George VI. So maybe some movies and some other things to watch to learn a little bit more about the history of the British royal family. So what kind of information can we get from pedigrees, and how do we determine whether a trait or a disorder is inherited autosomal dominant, autosomal recessive, or sex-linked recessive? So this is one some of you might be somewhat familiar with. It's a pedigree about PTC tasting. So PTC is this nasty chemical. Some of you maybe did this in high school, where you touch your tongue to some paper, and for some people it tastes really, really bitter, and for others it does not. And non-tasting, or the inability to taste that nasty chemical, is actually a recessive allele. And so you can see that indicated here in this pedigree. And so there are a couple of things that I just want you to note in this pedigree, and I have a couple of them, I think, circled in the next slide. So first, if we look at this pedigree, again, remember we're looking at... So in this slide, there are a few sample pedigrees for you to look at and practice. And I encourage you to try, look at some of the rules that we put in the notes, and look at each of these, and see if you can't come up with what you think is the most probable mode of inheritance for each of these pedigrees. And again, pause the slides and give it a try, because in the next slide I'll give you a few clues to maybe help you think about it. But give it a try and then move on to the next slide. So here in each of the pedigrees, I've circled a few important parents and offspring for you to pay close attention to as you try to figure out the most probable mode of inheritance. So we look at that first pedigree there. One thing I've circled is what is most likely if you have 2 unaffected parents having all unaffected children. So think about what would that be. Again, remember we're thinking about, when we talk about what's dominant recessive, we're talking about those that are affected here, the filled in symbols. So what kind of genotypes would both of those parents likely have if all of their children then were unaffected? And the other one that I have circled there is we also have 2 parents that are affected or have the characteristic or the trait, and they have a mixture of both affected and unaffected children. So looking at those 2 families that I've circled there, within this whole pedigree, what is the most likely mode of inheritance there? And if we look at the next one over to the right, I've circled where we have two unaffected parents and they're having mixed children. We have some that are affected and some that are unaffected. And if you look at the bottom pedigree on the right, you see the same thing. You see two unaffected parents having both affected and unaffected children. And then you also, in both of those pedigrees, you see it where one of the parents is affected and the other one isn't, and they have a mixture of affected and unaffected children. So be thinking about that and what that means for what mode of inheritance that might be. And then if we look at the bottom one on the left, I've circled a couple of things to pay close attention to here. So first of all, notice the top one that I've circled. We have an affected female, and all of her sons are affected. And then the other thing to note, of course, in this, the pedigree is fairly small, but you can see most of the affected individuals are male. There are two females that are affected, but it's not half and half. And again, you see an affected female having all affected sons. And then the other thing that you notice is when you have an affected female in that bottom family there, her father also was affected. And so be thinking about what the genotypes might be for each of these as you think about what the most probable mode of inheritance would be. And don't move on to the next slide till you've spent some time thinking about this. So in this slide, I've given for you what I think is the most likely mode of inheritance. So again, spend some time looking at these, looking at the rules that are in the posted notes on Canvas, and become familiar with the things that you would look for in order to determine what the likely mode of inheritance is.